Alright, Dr. McNeil, Alex Shaw behind the camera. We're here today to give you a brief overview and an appreciation for how transcription works in prokaryotes. So, gene structure, transcription in prokaryotes. Ready? Bacterial RNA polymerase. So RNA polymerase is an enzyme, writes RNA. Polymerase means it makes a chain, so polyRNA. And that is a DNA-dependent RNA polymerase. What does that mean? Well, it has to read the DNA and write a copy of the RNA. It's the DNA-dependent RNA polymerase. And these polymerases, when you hear me talk about it all the time, I'm not talking about a single protein that does a single job. We're talking about a multi-subunit complex. And like in DNA polymerase or RNA polymerase or telomerase, any of the major multi-protein complexes that we have, when you need multiple subunits to function, we call this the hollow enzyme. So it needs 6 different proteins to form the whole enzyme, the true enzyme. And that's the minimum number of protein functional units that can go together to write this RNA. Now, only one of those proteins actually writes the RNA. All the rest are required for specificity, speed, and accuracy. So it's important. So RNA polymerase, where does it go? How does it know where to go? Well, in bacteria, RNA polymerase finds a stretch of DNA, about 70 or 80 or 90 base pairs. That's not quite the same in eukaryotes. So we'll learn about that later. We call that area the promoter. I'm going to tell you a little bit about promoters in a little bit. And how does RNA pol know where it's going? How does RNA pol find the promoter? All right, it doesn't do it randomly. It does it through these other proteins called sigma factors. This is a lowercase sigma. Sigma factors are essentially interchangeable with RNA polymerase. So if you take one sigma factor and change it out for another, that same RNA polymerase will now bind a new promoter, a new place on the DNA. So any 2 sigma factors direct RNA polymerase to different genes. Okay? And bacteria are fairly simple, especially bacteria that just hang out in your gut, like E. coli, are fairly simple. So it has 7 main sigma factors. So basically 7 combinations of places where genes might be turned on. Okay? Genes are regulated outside of this, but this is the most basic level of where RNA polymerase knows where to go. Okay? So, what do genes look like? Well, in bacteria, like in eukaryotes, you have a double-stranded genome. Okay? I'm going to label the ends. Double-stranded DNA genome. And what's important? Well, the mRNA has to be exactly equivalent to one of the DNA strands. And we call that strand, in this case from 5' to 3', we're going to call this the coding strand. Why do we call it that? Because that's what codes the mRNA for a protein coding gene. Okay? The other strand is the template. Now, what's important about the template strand is that that's physically the strand that RNA polymerase will read in order to write an mRNA that looks like the coding strand. Okay? So if we're going to decorate genes, if we're going to decorate our DNA and show you what's in genes in bacteria, here's what a gene would look like. Well, we have a coding strand. We need to put the mRNA, you need to put the polymerase, we're going to cut that out. We need to put the bacterial polymerase in the right place. Okay? So I'm just going to blow this up real big. So what do you have? The first thing we have, part of the coding strand, is what we call the +1 site. This is also known as the TSS, or the transcription start site. The TSS. Okay? That is the physical place on the DNA that encodes the very first nucleotide of mRNA. Okay? But remember, this is now decorated on the coding strand. In reality, the RNA polymerase is going to read the template strand to write this. Okay? What do you have next to that? Well, for a while, you have DNA that will be transcribed, but it's not going to be translated because we haven't reached our translation start site yet. So this is what we call a 5' UTR. And UTR is very simple. It's the untranslated region. Okay? Next to that, in bacteria, we have a protein coding gene. So I'm just going to write gene 1. Okay? Bacteria are special in that one mRNA can code for multiple proteins. So next to that, I might write gene 2. So now, with one transcription event, we'll turn on 2 genes, right? It turns out, when these 2 genes are all controlled by one transcriptional event, you call this an operon, right? Operons functionally work what I'll call in the same industry. And we'll learn about a few of these. But what does that mean? What does working in the same industry mean? It means if we need to metabolize a certain sugar, maybe this protein encoded by gene 1 imports the sugar into your body, or the E. coli cells. Maybe gene 2 modifies that sugar so it can go into the glycolysis pathway. Okay? So these things, these 2 genes encode proteins that do a job that coordinates with each other, that you need them both on at the same time. Okay? So, the other thing that comes out of this now is any mRNA that comes from an operon. mRNAs encoding more than one gene are going to be called polycistronic. Poly for many, cistron means gene, many genes. Okay? So, what's after gene 2? Well, let's say this is a 2-gene operon, and so now all that's left is what on the DNA codes for the 3' untranslated region. And then there's going to be some kind of coded termination signal somewhere in 3' UTR. Okay? Check out the videos on transcription termination in prokaryotes to figure out what these signals are.